Welcome to the presentation of our paper with the title Convolutional Kernel Networks for Interpretable End-to-End Learning on Multi-Omics Data. In this work, we investigate the possibility to create inherently interpretable prediction models for tabular omics data utilizing kernel networks. What motivated us to do this research is the fact that in healthcare and computational biology in general, the size of available dataset ranges from small to large scale. One model type that can successfully apply to all these different scales of dataset sizes are deep learners. Due to efficient training procedures, they can be applied rather efficiently to large-scale data, but also be robustly trained on smaller-scale data. And due to being highly over-parameterized, they offer very high prediction performances. However, they usually are deployed as black boxes, which means that they do not offer insights directly into the decision boundary learned by models. So we ask ourselves, is it possible to create inherently interpretable models, so-called white boxes, that can efficiently scale to all different sizes of datasets and also offer the expressiveness to achieve high prediction performances? And we tackled this research question by using kernel networks, an approach that combines kernel methods on the one hand with neural networks on the other hand. So, how can you combine kernel methods and neural networks? To recap, a kernel method compares data points within a potentially infinite dimensional Hilbert space. To make this comparison computationally feasible, they utilize the kernel trick, which allows them to compute the similarity of data points within these infinite dimensional spaces without explicitly projecting data points into the spaces. However, neural networks learn in a way that they explicitly project data points from one space into another. We cannot feasibly project data points onto infinite dimensional spaces within a computer. So how can we incorporate learning within the Hilbert space of a kernel function into a neural network? And the solution was proposed by Mayral in 2016, and he proposed to use a variant of the Nystrom method. In this variant, we first select a set of anchor points. These lie within the input space and can be selected, for example, with simple k-mean clustering, and use these anchor points to span a subspace of the infinite dimensional Hilbert space. We furthermore can use the anchor points to define an explicit parameterization of an orthogonal projection from the potentially infinite dimensional Hilbert space onto the finite dimensional subspace spanned by the anchor points. This orthogonal projection preserves the distance between data points in the original space, which means that two data points that are close together in the original infinite dimensional space are also close together in the projected finite dimensional subspace. This preservation of distance secures the very fact that allows kernel methods to learn, and this method allows us to utilize learning within the Hilbert space of a kernel method into a neural network. The method was originally proposed to offer robust trainable neural networks for small-scale image data. However, we investigated how a specifically chosen kernel function can lead to inherently interpretable kernel networks for tabular omics data. And the kernel function we selected to utilize in our kernel networks is the pathway-induced kernel. This kernel computes the similarity of two molecular measures based on the structure of a network. It's defined in the following way. So for two inputs, xi and xj, which are just tabular omics data, we use then a Laplacian matrix to compute the similarity of these two measures. A Laplacian matrix is just a matrix that encodes the structure of a network. And this Laplacian is the point where we introduce prior knowledge into our kernel function by utilizing a biological meaningful network, for example, an interaction network. We can further increase the expressiveness of this kernel by not taking the whole interaction into account in the function, but actually splitting the network into meaningful subparts, for example, subparts that correspond to pathways. Doing that leads to a kernel function that actually is a set of kernel functions, with each function corresponding to a pathway-induced kernel. Here you can see where the name pathway-induced kernel comes from. So this set of kernel function was utilized by us to create our kernel network. And with our paper, we are introducing two different architectures for a kernel network. The first one starts by utilizing the pathway-induced kernel. It takes the input, which is a molecular measure, and then projects parts of the input that correspond to certain pathways onto the finite dimensional subspace spanned by the anchor points of the corresponding pathway-induced kernel functions. We then utilize max pooling to create a single activation per kernel embedding. These kernel activations are then combined using a linear fully connected layer similar to a multiple kernel learning approach. Since the fully connected layer is ensured to be linear, we can then use the weights learned by the network to get an insight into the decision boundary learned by the model, similar to how you do this in multiple kernel learning. This insight into the decision boundary is also called a global interpretation. The second architecture proposed by us starts the same way as the first by utilizing the pathway-induced kernel to project parts of the input onto this different kernel embeddings. However, in this architecture, we then treat the kernel embeddings as instances in a multiple instance learning problem. So we create a bag of instances and utilize attention-based MIL to compute attention rates for each instance, which in turn are then used to create an ensemble of the bag of instances that is used for prediction. These attention rates have the very nice property that they get computed for each input individually, which allows us to use them to get an insight into a singular decision, a so-called local interpretation. We were interested in how well our models are able to perform on healthcare-related prediction tasks. So we decided to evaluate the prediction performance capabilities of our comic models by utilizing a survival prediction on breast cancer cohorts. We selected 6 different publicly available breast cancer cohorts that provide gene expression data and shows as an endpoint either distance metastasis-free survival, DMFS, or relapse-free survival, RFS. Furthermore, we converted this continuous endpoint into a classification by selecting the commonly used threshold of 5 years, which means that patients were separated into a class of patients with a DMFS or RFS of more than 5 years and patients with a DMFS or RFS of less than 5 years. To create our models, we utilized the 50 hallmark gene sets, which are gene sets that correspond to biological meaningful concepts, for example, pathways. We evaluated the performance capability of our comic models by utilizing 10 times tenfold stratified cross-validation on each of the 6 different cohorts. So what you can see here are the AUC value on the validation splits for the stratified cross-validations on each breast cancer cohorts. In yellow, we show the results for our comic models, and the results suggest that Comic is able to perform similar or outperform to state-of-the-art methods that also utilize pathway information to make a prediction. While these performance results were very promising, we also investigated the interpretation capabilities. Keep in mind, for the following results, these are directly extracted from trained models that were trained using a simple end-to-end learning, utilizing atom optimization and class balance loss. We did not perform any post hoc optimization or utilize post hoc modeling to create interpretations. So the first interpretation that we investigated was the global interpretation. What you can see here is on the x-axis the 50 hallmark gene sets or pathways, and on the y-axis the weight learned for each pathway. Furthermore, the box are created by using the weights learned on each of the 6 different breast cancer cohorts. And the first result that we saw was that the pathway weight signature remains rather stable across cohorts, which was expected, and it's a very good first result. Furthermore, we saw that high absolute weights correspond to pathways that are known to be associated with breast cancer. To just show you a few, we see a high absolute weight for androgen response, which is associated with breast cancer pathology and progression. Also, we see hedgehog signaling associated with tumorigenesis, progression, and as a therapeutic target. Furthermore, we see a high absolute value for Notch signaling, which is associated with progression, and also MYC target V1, which is associated with cell proliferation and immune suppression. This shows that the global interpretation provided by Comic models is actually biologically meaningful. To investigate the local interpretation, we randomly selected one of the 6 cohorts and then retrieved 3 random patients from this cohort. Patient 1 was a patient that was correctly classified to have a DMFS of more than 5 years. Patient 2 was correctly classified to have a DMFS of less than 5 years, and patient 3 was a patient that was wrongly classified to have a DMFS of more than 5 years, and the true DMFS was less than 5 years. We then retrieved the attention rates calculated for each of the 3 patients and plotted them as heat maps, which is shown on the right side here. The x-axis again are the 50 hallmark gene sets, and the color indicate the value of the attention rate corresponding to the pathway. The first result that we immediately see is that there is a clear distinction between the 2 classes in the signature of the attention rates. Furthermore, we see that the patient 3, which is wrongly classified to have a DMFS of more than 5 years, has a very similar attention rate pattern compared to patient 1. The difference in color just occurs due to the difference in the scale, as you can see on the right side. This pattern was investigated by us in more detail in the supplement, and we saw that while some patients that were wrongly classified can be clearly identified to be wrongly classified due to their attention rate pattern, we have a lot of patients that show a very similar pattern to patients in the class they get assigned to, which could point towards a mechanism for these patients that causes them to have a DMFS of less than 5 years that is not actually learned by the model. In any way, this suggests that there is a group of patients that should be investigated in more detail. In general, the results seen for our local interpretation capability suggest that Comics local interpretation provide meaningful information and enriches the prediction made by a model. So to conclude my talk, we show that combining kernel functions with neural networks can lead to inherently interpretable models for tabular omics data. We furthermore show that our Comic models perform either similar or outperform state-of-the-art prediction methods that also utilize network or pathway information. And finally, we show that the global and local interpretation capabilities offered by comic models are actually containing biological meaningful information. In the corresponding paper, we show further results. For example, we show how you can extend comic models to multi-omics data and also show results for multi-omic data prediction tasks. Furthermore, we show that comic training scales linearly with the number of data points and omics modalities, with results that suggest that comic models can be efficiently trained on datasets with more than 100,000 data points. And with that, I would like to thank my two co-authors, Bernhard Reuter and Nico Pfeiffer, and also the whole Pfeiffer Lab for very fruitful discussions and support. And thank you for your attention.